hope all of you have downloaded this app.
Yeah. So you can open this out, but let's see what we do with it. But before that, actually, I think my yesterday's talk was a little bit fragmented, and going by the questions that I was getting, I felt like a lot of these points are not coming together for you guys. And what I was trying to do was trying to link some of these acoustics component to other things that you had heard through the day. So let me just recap quickly. The first was that most acoustics is like most of the, when you think about avian acoustics, most of it is vocal, but there are exceptions. So I intended to start with the exception. That is what you heard, like the feather and stuff like that. The other thing is that you heard about the concept of sexual selection, and a lot of the songs, the bird song. A lot of the bird song work is in the sexual selection framework. This we talked about, but what I did not specifically mention was that the acoustic adaptation hypothesis, the different habitats, that is actually another natural selection framework. Okay, so you have these two operating: the sexual selection, where the females are selecting a specific kind of songs, and then you have the natural selection, where there is some adaptation to a specific kind of habitat. Okay, so these are two kinds of pressures that acoustic communication kind of faces. And the dialects in birdsong can actually result from either of this. So it can be because of sexual selection, or it can be because of natural selection. So this is something to bear in mind. I also briefly touched upon the idea that bird songs, birds in a community have differentiation in bird song. So this is also something to keep in mind, that when you're comparing a bird from a specific community with that bird in another community, that may also be some kind of an adaptation to occupy a different acoustic space. It need not be in the sexual selection framework. So when you're comparing songs, these are things that you need to keep in mind. Okay. We also talked about alarm calls, where there is a convergence of signals, different kinds of species, producing very similar calls. And we talked about how there is a phylogenetic relationship in how close or similar songs are. So more related species can have similar songs. Yeah, I think this is something that I wanted you all to get. So this this module is largely about techniques, about how you would go about conducting a study in, let's say, vocalization. But first, before we do that, so what I want you all to do is actually open out that spectrogram viewer software. Okay, having the app. And what I'm going to do is I'm going to play a certain frequency from the other app that I sent you. So make sure that you don't start that app, because that's going to emit some sound, and we don't want everybody to be emitting different sounds. So is the app working for any of you? Yeah. Yeah.
Yes.
So we are at about 1700 hertz. So if you see, there is a button called scroll on the left, you can hit that at the bottom left. Yeah. So that's 3400 hertz, that's about 3 kilohertz. Yeah, that's about, right? Okay. Ignore that. So, can you hear anything now? It's actually really high. Right? So, you saw that the frequency was getting cut off when I went, that was at 12,000, and this doesn't allow you to record 12,000. Okay, so you can actually play around with these quite a lot. So what I'm doing now is I'm producing two sounds at the same time. Yeah? Yeah, can you see that there are two frequencies that are there, right? So I think, so basically I just wanted you to kind of get an idea of how to visualize sound. Because when we're talking about spectrograms, it should not be something that is abstract. So basically there are these different frequencies, and we talked about spectrograms yesterday. Okay, and what you saw was a live spectrogram. I mean, in fact, if your app is still on, it will show you, the where the human sound falls and things like that. So you can play around with this at tea break, and you'll see that the sound creator, the frequency generator app actually has a lot of features. It can be quite a lot of fun. Okay, so bird song. How do you study bird song? So the first thing I want to actually stress, although you will have a module on study design, is that the most important thing, as with any, I expected that. Yeah. The most important thing to remember is the question. Like, what exactly do you want to study? Like, what is it, what is the specific biological question that you're trying to answer? So that is an important thing to consider at the outset. And then you definitely need some information on natural history of the species. So, suppose you just want to study some random bird, I don't know, Jerdon's courser, let's say, okay? And you don't know what it does, like you have very little information about its natural history, then you will not be able to kind of, figure out where to go to record the bird, where would you expect to find it, how long does it, call, in case of the courser, or if it's another songbird, how long does it sing. So a lot of the study design factors would actually come in from understanding the source. So whatever is creating the sound, okay? So that's really important. So good natural history is often, that is actually the key to having a good study design. And the study design by itself is actually very important as well. And what that means is that, so some of the things to consider with study design, we talked about how songs can vary very quickly and because of different things. So whenever you're studying something that has so much variability, you need to have a design that controls for this or can account for this variation in some way. And that you can do by having sufficient samples between individuals, because you know that there is a lot of variation within individual and between individuals. Okay, and if you're doing a study across populations, you need to be sure that the variation that you're looking at, any difference that you're looking at, is over and above an individual variation or the group variation. Okay, so all of this you need to consider before you start the study. Absolutely, the worst thing that you can do is record some sound first and then think about what can I do with this sound. Now that I have this recording, what do I do with it? Most often, that is not going to get you anywhere. So, I mean, unless it is just something for, uploading on eBird, or, you put up on one of those websites just as a record, it doesn't by itself serve any specific research question. So, just because you have a recorder, don't record. So record when you have a real question and a real study design. Okay, so this is something that you have to really keep in mind. Otherwise, you'll just go berserk with all the recordings and you won't know what to do with it. So now coming to the real part, I think. So how many of you have done some acoustic recording? Okay, quite a few have. So that's good. But anyway, these are some basic recording gear, and I just want to quickly acknowledge that this is stuff that Viral Joshi, one of my students, put together. So these days you get different kinds of digital recorders. Zoom has very good options. You can look at those. It's not very expensive. Then what most field biologists use today is something called a shotgun microphone. And the microphone usually has a windscreen. So this is just so to avoid, the sound from, right? Okay. And you can also, you should always have headphones. And then it's a little bit of it is your individual preference. If you have, like one of those big headphones, and if you're working in, let's say, elephant country, it's probably not a good idea because you won't hear anything else. So you also cannot localize. If you're just only listening through with the headphones, you don't know where the sound is coming from. If your target organism moves a little bit, you won't be able to figure that out. So the best thing to do is kind of leave one ear on and one ear off, so that you can kind of at least listen to something that's happening outside. So there's a whole lot of other things to be considered on the quality of recording, because there is a technique for recording, but also various kinds of equipment. For very high quality recordings, what people do is use a parabola. And what the parabola does is just like a dish antenna, it just collects the waves and just directs it to the center, right, to the focal point of that reflector. And you have a microphone over there that collects this. So what happens is that you have very clear recordings when you have something like that. So this is the gold standard. Okay, so if you're just working on one species, you know that the species is here, then this is what you need. And there are different kinds of parabola. You can see that some of the early parabola, and this is Viral, and he's very famous because I think he has about 800 species of birds recorded. And a lot of it he did with this, this cement mixer kind of a thing, turned into a parabola. I mean, I don't think he himself recommends this anymore, so don't try that. But the point is that it is possible to do things like that. But I would still say that parabola and using parabola for recording is a really advanced step, and you may not need that. Most often a shotgun microphone is probably the best on field. It's really portable, very easy to use and carry around. I think a question a lot of you may have is, can't I just record on the mobile phone? I mean, I have it with me, I can hear the animal, I mean, what's wrong with that? This is a little bit of a tricky thing. I mean, there are some things that work well and some things that don't with mobile phone recording. One of the important things to remember is the file type. So most often the default recording is an MP3, and that simply doesn't work because, as you can see, it's a really the idea of MP3 is kind of how the data stored is in lower file sizes and it samples very less compared to a wave file, which you can see on the right side. There's also something called the sampling rate, which you can set on your recorder. So what the sampling rate is, just like if you're doing anything in GIS, you have a map, and that map has a resolution, right? This is something very similar. So what you're trying to do is determine what is the resolution at which you should record. And there's a simple kind of rule for this, which is that you look at what is the highest frequency of your target organism. So let's say, now you have a fairly good idea of how different frequencies sound. So if the maximum frequency is about 10 kilohertz, then you can set your sampling rate as 20. Okay. So usually there are some presets in your recorder, and it's usually 22, 44, and so on. So you can choose that. But something to note here is that you should try and keep the sampling rate constant. So let's say that you're interested in looking at a particular species for which you kind of set the sampling rate at 22 kilohertz. But then suddenly you got interested in some other species, and then that you record at 44 kilohertz. And if you want to at any point compare these two, you'll be stuck. You can do it, but it's a real problem. So it's better to kind of fix on the sampling rate initially. And all of this boils down to the first thing that I was talking about. It depends on your question. So it depends on your target species and what you're really doing. So depending on that, you can make your choices. So here I just want to say, so mobile phone recordings, maybe not for kind of research grade stuff, but if you're doing a recce and you don't want to carry a recorder along, I think that if you can get your phone to record a wave file, you'll find an app. There are several apps today that can do that. And then you can do this, really cool thing, which is get an XLR adapter and you plug your shotgun microphone into your phone. And this can give you quite good recordings. It's not, you may still have some limitations, but some preliminary recordings can be done in this way. So, having recommended this, I don't know if this is really practical, because your phone is probably more expensive than the recorder. So today, recorders by themselves are not expensive. It's a microphone that's expensive. So so you can use your phone, but you'll probably get not great recordings. So yeah. And finally, you can also use passive acoustic recorders. Again, it depends on the question. Things that you can't get from passive acoustic recorders is you don't know which individual, you don't know what the individual is doing, so the behavior, all of that is lost. So the question then becomes actually slightly different if you're using a passive acoustic recorder. Okay, so I think that so that was about acquiring data. Once you acquire data, I'm sure all of you know of various softwares to kind of edit sound data, but Raven is pretty much the best today for all research grade analysis. Have any of you used Raven before? Okay, quite a few. Yeah. So Raven is free, you can download it. It's developed by Cornell Labs. I have to say that, so this is not, I'm not really going to tell you about how to use Raven here, because that we have a full workshop actually for something like that. And you should just look out for some of those. We conducted one last year, and hopefully we'll do again sometime next year. But I will tell you a little bit more about once you have the spectrogram in Raven, you can then measure different variables in the song. And these are, these are very simple measurements. It's just along the x-axis, which is time, and then the y-axis, which is frequency. And what you have to actually do in Raven is, if any of you have used GIS, you digitize maps, right? Like you create a polygon around different shapes, different forests and other things. So very similarly, you have to draw boxes around each of the nodes that you see. And with that, you will get data on minimum frequency, maximum frequency. So this, there's low frequency. So if you draw a box here, you get the high, you get the low, and you get the bandwidth. And similarly, if you have a box drawn this way, you get the starting point and the end point from which you get the length. Yeah. So these are some of the common and the simplest parameters. Raven also has a lot of complicated kind of parameters, and some of them are to do with where energy is. So you can have a node like this, where let's say that a lot of energy is here, and this is just a faint little trail. Maybe that doesn't matter. So how do you know what is important? So for that, the Raven folks have a really nice, additional metrics that are there under the hood, and you can explore those. It divides basically each node into 4 quartiles, and then it looks at where energy is more. And so this is not very intuitive. So I don't really use it unless there is something specific I want to do with it. For all, let's say, variation of populations and individuals, this is pretty much good enough. That is what we work with mostly. Yeah. And finally, if you want to start out, there are a number of sources that are there to get your data. Not to get your data, to get some kind of data. I think everybody knows Zenocanto?
Yes.
Yeah? But there is a problem. Most of the Zenocanto recordings are MP3. It's very easy to access, but it's all MP3. So if you want to use it for some research, it's going to be a little tricky. That is one. Then you have Macaulay Library that is growing very rapidly, largely because of the association with eBird. And this has WAV files, but one of the problems is that you need permission to download some of these files. So it's a small step kind of a thing that's there, but you can get that. Then there is Everset, which is Pam Rasmussen's. This has WAV files too, and it's easy to download, but there is only a limited data set. What that means is not all the species are there in the database. Only whatever Pam Rasmussen and a few others have, only that's there. Yeah, so we'll just move on to some molecular lab techniques. Obviously, this is also not really going to go do much justice to the whole idea, but we'll give you some, some overall understanding of what happens. Has anybody done a PCR? Everybody? Sorry, can I just see a show of hands? Like just anyone who's done a PCR? Okay. So quite a few of you have done a PCR. So it's, all right. You can Google that also, but I'll tell you again. This is like a standard slide actually. That is, I copied it from the acoustics page. But the point is that before you start doing any molecular lab work, you need to have a question, which is to figure out why you want to do this. What is the big biological question that you're trying to answer? And it may not seem like it is important, but the natural history of species is actually critical, because you may get a whole bunch of genetic data and some patterns, but you won't know what that means. So that makes sense only in the light of some actual species biology. So there's no point in collecting genetic data if you don't understand the species ecology. So it makes sense only if you can kind of connect it back to what the birds are doing, right? So I think that's something that is very important. So I would say, never start out with just the genetic data as a starting material. Start out with the bigger question as a starting point, and then use genetics to answer that. And once again, I think that not many people think about study design as being important, but actually it is very important, especially with genetics. So where you collect your data and how many samples you collect, these have a big bearing on what your inference may be. So this is again something that has to be considered carefully. Sorry, is there a question, Jobin? Oh, okay, okay, yeah, just stretching, yeah. Yeah, okay, so so study design is let's say very critical, because to give you an example, are you actually looking at differences, between population that are just between individuals in a population, or are you trying to look at differences across species? And depending on this, primary question, the kind of data that you have to generate, the numbers of samples that you have to collect, all of them will change. Yeah. So one is more towards population genetics, and the other is more towards phylogenetics. So these have big consequences on your downstream analysis pipeline. So once again, study design is very important. And the worst thing you can do is collect some sample first and say, okay, I have the tissue of some species. What can I do with this tissue? Like, what should I do? I have this genetic data, like I have the sequence data. Now, what can I do with this? I think that is like, you can imagine that this is pretty bad. It's as bad as saying that, okay, I have some point count data from here and there. Now, how do I fit this into something? And it's always a bad idea. So it's better to kind of have all of this sorted out before you get to the genetic data. Yeah. So you've not Umesh hasn't given you the bird capture thing, right? It's coming up. Okay, yeah. So you're going to see a little bit of the details on bird capture techniques and so on, but this is my collaborator Vishnu. What we do is we put up these nets and we catch the birds and we take a drop of blood. You don't have to collect blood. You can also get any kind of other material. People have taken buccal swabs. Yeah. Feather. Yeah. Problem is that all of these have chances of more degraded DNA. So high quality DNA is still in tissue or blood. And tissue should be actually fairly easy as well, but the problem is it's more difficult to get permits for them. Blood is easier. Yeah. So you can collect blood in something called a lysis buffer, and that's how we do that. And this stays in room temperature. You don't really need to put it in the fridge right away. You can also put them on FTA cards, which are special kind of blotting paper, and that also stays at room temperature because of some proprietary chemicals that are there on it. But it's much more expensive. So I'm going to skip a lot of material of what happens from when you collect the blood sample to getting a sequence. So, but I will explain just briefly. Once you get the sample, you bring it to the lab, and then you extract the DNA. Basically, you break up the cell, and then you kind of only want the DNA from that. So, once you do that, then you can send your sample for sequencing, mostly at some facility. And the most common and the simple sequencing pipeline is the Sanger sequencing, which just gives single reads. Okay, it's a very old technology. Again, there are lots of videos. You can just Google that and look at how Sanger sequencing works. And I can also try and post that, in your reading materials so that you can actually look at it later. So, what happens after the Sanger sequencing is that, so I think everybody knows that you have A, T, G, and C, right? That's what you should get back. And you get a file which is like this, which gives basically, this is the fluorescence, it's the readout from the Sanger sequence. And you have a forward and a reverse. So let's say this is the forward and this is the reverse, the two strands. And there are softwares that could stitch this together, and you want a consensus. So for every base, basically you want to make sure that it's T at that same spot in the forward and in the reverse. So if you have very good quality sequence, you'll see these nice sharp peaks, as you see here. And you see here, this is poor quality sequence. Yeah. Okay. And then the other thing is that Sanger sequencing can only do about 700 odd base pairs of length, and often that is not enough. Even if you want to get cytochrome B, you need 1,000 plus base pair. So what most people do is that they stitch it together. So they do two reactions. So you have a part 1 and a part 2, and then you stitch that together. Yeah. So essentially what happens then is that you have 4 reactions and you get one sequence. So at the end, what you're getting is a consensus sequence of about 1,500 base pairs, which is put together from these 4 different reads. Yeah? Okay. And once you have this, then, there are 2 approaches, and I'm going to only show you the simplest one, which is just building phylogenetic trees. And I'm not really going to go very deep in that. The first thing is that, do you remember Anand's talk? He also spoke about phylogenetic trees, right? So, like what I'm talking about is building trees with this ATGC. And here these are considered as some characters. But can you think of building a tree with some other trait? Some other character?
Spectrogram matching.
No. What I mean is, are there any traits? Are there any characters other than ATGC that can be used to build, to look at relationships? I think you're getting confused by my question.
Proteins. Amino acid sequence.
Yeah. Yeah, you can do it with morphometric data, right? You can basically you can look at it's just instead of A and T or G or C, it's any other character. Only thing is that you need to know how these characters change from one to the other. So anyway, I think so here what we have is sequences which are not aligned. So 3 different species. Yeah. And what's done here is that you've created an alignment, and you've done that by introducing a little gap here. So there are softwares that do it. I'm sure some of you already know these. But essentially you do this only so that you get homologous characters. So that you're comparing the same thing with the same thing. So you're comparing the arm with the arm, right? So this is an important step. And more the characters, the better your relationship, the inference and relationship is. Yeah? So I think that's pretty straightforward, right? Because if you have only one character, maybe your 2 groups that you're comparing with are not different by that character, but there are several other characters that they're different by. And this is the same thing that happens with genetic data also. So you may have a sequence of 5, like I showed you 1,500 or 600 base pair, but the informative sites where there is variation, that may be very little. Like for example, at this point it's A and A here, and C is different, right? So that is informative. So that is one point at which it differs from the other, and you can then use that information to kind of separate these 2 groups, right? So according to that, these 2 will form one group, and this will be different. And you could go on and do this. So if you are here, all 3 are the same. So actually that doesn't add much information. You come to the next one. These are also the same. So again, there is no information. So your information only comes from any variation. Yeah? So these are called like some kind of polymorphism. So you may have heard of the term SNPs, single nucleotide polymorphism. So that's how that works. So essentially we are looking at anything where there is some variation, and that is then used to stitch together relationships between different groups. I'm not going to go through a description of how to exactly build trees, but I do want to just talk about this thing about which relationships can actually be trusted. It shows you some relationship, but you don't actually know what the relationship is because there is no statistical data associated with the power. And you do that by doing a bootstrap analysis, and what you have to see is at the node you look for the value, the bootstrap value, and if it is generally under 95, people don't think it is a very it's something that you can trust. So I would say, if you have a tree, just look out to make sure that your bootstrap support is high for every node before you make any inferences of actual relationships. The other question that people often have is how much data should you have? How much data do you need for answering your question? And there is no simple answer for that, and there are different technologies also. We talked about Sanger sequencing. So in Sanger sequencing, you have this single strand, and then you have to kind of keep generating these single strands which are independent, so that you're getting actually information from different parts different nuclear, mitochondrial, and so on. So usually people target multiple genes that are independent, and then you kind of put them together in different analytical framework. But more recently, next generation sequencing techniques have become more common, and in that there are three major kind of ways that people have been working, especially with birds. So this DD-RAD, it is a it's a double digest kind of a method. What it does is it just basically chops up your entire genome into really small bits, and then it pulls out SNPs from all across the genome. But it is still reduced. It doesn't give you an entire genome, only small bits from different parts. And often you don't know which parts are coming from where unless you have a reference genome to map this to. So there are some limitations to using DD-RAD data, and because you use some particular methods to chop the DNA, every time you do this, the chopping is random. So you may get different, chops, literally. So, so comparability between data sets is poor with something like DD-RAD. So UCEs are very interesting. These are ultra-conserved elements, and these are areas in the genome that are conserved across different groups. So there is a vertebrate UCE, and that can be used for birds, it can be used for mammals, for any vertebrate. So that part is conserved all across all vertebrates. So it's like a primer that sits there, and then you can sequence, on either side of that using any of the next generation sequencing platforms. So you get a read that is conserved in the center, and as you go away from the center, it becomes more variable and you start getting these SNPs. But the cool thing about this is that there are 5,500 probes in one set, 5,500 primers. So you get a lot of data. It is still not the entire genome, but it is still quite a bit of data. And you get a lot of SNPs, so you can get more data and more, more information about the relationships that you're trying to look at. And of course, the big, the best you can do is to get the whole genome itself. But even with whole genome, there are various levels of coverage and so on. So it's not really very straightforward. There's a lot more in that. But I think that ultimately, all of this depends on the question. So this bird that Umesh talked about in the biogeography, the short wing, which is now Sholicola. So when I worked on it in 2010, I think we used 4 genes, and we found a particular pattern, which is what Umesh was describing across the Palghat Gap, about 4 million years or 5 million year old divergence. It was a different species. Later we added I think 8 more genes to that. That pattern did not change. Then we added microsatellite data to that. That pattern did not change. And later we have added next generation sequencing data to that, and that pattern still doesn't change. I mean, that is simply because it is such a very clear division. And others have sequenced the same thing, and they also find the same kind of dates and divergences. So if you're lucky, your organism is just very clean and straightforward, very different, and you can detect that even with a 600 base pair, length Sanger sequence. So it really depends on what is happening in the system and what your question is. You don't really need to hit it with the most intense, data-driven method that is there. because some simple questions can be answered with very simple data. So I would say, start with something simple, and then if your question does turn out to be complicated, if the pattern is not very clear, then you can add more and more data to it. I think that would be the way to kind of work on this. That's about it. Any questions?